Hello, everyone. Welcome to today's live broadcast, Harnessing CAR-T Therapy for Hematology and Solid Tumors, Experience from City of Hope. I'm Mike Christel, Managing Editor of Applied Clinical Trials, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by ACT and sponsored by Precision for Medicine. I would like to share a statement from our sponsor. Precision for Medicine is an accelerator of innovation devoted to supporting the development of new medical treatments and breakthroughs. As a precision medicine clinical research services organization, Precision has brought together new technologies, expertise, and operational scale to help the life sciences industry improve the speed, cost, and success rate of bringing life-changing therapies to patients. We have a few important announcements before we begin this webcast. This webinar is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Should you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the dock at the bottom of your presentation window. I would now like to introduce today's speakers. We are pleased to be joined today by Dr. Elizabeth Budde, Dr. Saul Priceman, and Dr. Al Blunt. Dr. Budde is an assistant professor in the Department of Hematology and Hematopoietic Cell Transplantation at City of Hope. Dr. Budde's academic and clinical interests include investigating and designing novel means of immunotherapy for patients with lymphoma and leukemia, and she is part of the T-Cell Therapeutics Research Laboratory, investigating the use of CAR-T cells to treat blood cancers and other diseases. Board certified in medical oncology and internal medicine, Dr. Budde has received many honors and awards for her work. She was chosen as a special fellow and clinical research awardee by the Leukemia and Lymphoma Society. She was a recipient of the Edson Immunotherapy Pilot Award from the J. Orrin Edson Foundation and is an awardee of the Stop Cancer Foundation Seed Grant. She is also a recipient of the prestigious Damon Runyon Cancer Research Foundation Clinical Investigator Award. Elizabeth has served as the principal investigator on several innovative translational research protocols and has published her work in many peer-reviewed journals and given presentations at national and international meetings. Dr. Priceman is assistant professor in the Department of Hematology and Hematopoietic Cell Transplantation and associate director of Translational Sciences and Technologies in the T-Cell Therapeutics Research Laboratory. Saul is a principal investigator and provides scientific oversight on solid tumor immunotherapy programs focused on the development of CAR-engineered T-cells and rational combination approaches, prostate cancer, breast cancer, brain metastasis, ovarian cancer, pancreatic cancer, and other solid tumor types. Last but not least, Dr. Blunt is Senior Vice President, Medical at Precision for Medicine. He has more than 20 years of drug development experience with a focus on oncology trials, and at Precision, Dr. Blunt is responsible for providing strategic guidance, oversight, and consulting for the design, conduct, and analysis of cancer trials. Al has provided global leadership for Precision's team of oncology medical monitors. His experience includes clinical trials from phase 1 to phase 3 and for a wide variety of anti-cancer drug classes and cancer types. Thanks, everybody, for joining us today. Elizabeth, please get us started.
Hello, hi. I'm Elizabeth Budde from Department of Hematology at City of Hope National Medical Center. Today, I will present CAR-T cell therapy for hematologic malignancies. And here are my disclosures. CAR-T cell therapy is no longer an unfamiliar term nowadays. It is a cell-based gene therapy. It works by harnessing the power of the immune system to recognize and eliminate cancer cells. It involves in genetic modification of an ordinary T cell and transform the ordinary T cells into a super T cell, which can recognize the cancer cells at the same time it will clone itself because it's a living drug. Building on the success of CD19 CAR-T cells in B-cell lymphoma, globally we have seen an exponentially increasing numbers of CAR-T clinical trials. from 1 in 1997 to more than 1,000 in May 2020. Majority of our trials are devoted to patients with hematologic malignancies. The most common one is lymphoma, followed by acute lymphoblastic leukemia, followed by myeloma. AML only represents 40 of the trials, which I will mention later. At CAR Hope, we have already treated more than 500 patients, with the first patient entered clinical trials in 2000. And by May 2020, we have treated more than 500 patients. And all of them received one of the three categories. They can either receive CAR-T cells from FDA-approved products. They can also receive CAR-T cells from CAR Hope sponsored clinical trial using CAR Hope manufacturing products. And third, they can also receive a CAR-T product by enrolling onto a pharma clinical trial open at CAR Hope. So let's go to an FDA-approved CAR-T cell product. The first FDA-approved CAR-T product is T-cell genaglucel for patients with acute lymphoblastic leukemia. This approval is based on the results from the pivotal trial, ALIANA trial. In this clinical trial, patients aged 3 to 21 were enrolled onto this CAR-T study. 79 patients received the infusion using their own CAR-T product. As you can see here, complete remission rate is exceptionally high at 82%. Majority of them had MRD negative disease. With a median follow-up 24 months, 45% of them still have ongoing response. The relapse-free survival at 18 months is 66%. And based on these results, the FDA gave approval for this product to be given to patients with pediatric or young adults and ALL. And if you notice here, only 8 patients with complete remission move on to allogeneic stem cell transplant. All the other responders did not receive further treatment. In patients who relapse after CAR-T therapies, 75% of them have CD19 negative disease, and only a minor population of them has CD19 positive disease. Recently, Schwartz et al. Summarizes their real-world experience using T-cell genaglucel using a CIBMTR registry. In the real-world experience, patients with less bone marrow burdens, 44.7% of them has less than 5% of blast in the bone marrow, and 31% of them has allo transplant, and 11% of them has extramedullary disease, which are less than 76% in the pivotal trial. When we look at the response rate, in the real-world experience, 88% of patients, 100% of them had achieved minimum residual disease negative response, and the 6-month duration response is 77%. So longer-term follow-up still needed to demonstrate the durability of the response. Nevertheless, in the real-world experience, there seems to be a more favorable safety profile with only 13.3% of CRS and 8.6% neurotoxicities. This might be due to the improving management of care for these patients. For B-cell lymphoma, 2 products are approved. One is Yescarta, Axi-cel, which was approved on October 18, 2017. Kymriah T-cell genaglucel was also approved on May 1, 2018 for similar patient populations. Just to mention that Axi-cel is also approved for patients with primary mediastinal B-cell lymphoma. These 2 approvals are based on results from 2 pivotal trials, ZUMA-1 and Juliet. In both clinical trials, you can see the CR rates are as high as 58% in ZUMA-1, 40% in Juliet. And the ongoing CR rate with the median follow-up 27.1% months in ZUMA-1 is ongoing. The ongoing CR rate in the Juliet trial is 37% with a median follow-up 15 months. Both trials, patients actually have experienced significant second-release syndromes and neurologic toxicities. Recently, 2 presentations looking at the real-world experience using Axi-cel were presented at ASH 2018. Both manuscripts were recently accepted by JCO and will be coming out soon. In both of the studies, patients in the real world, 61 in the first study and 58 median age in the second studies were comparable to the ZUMA-1 patient age. And also, if you noted, in the real-world experience, more than half of the patients received bridging therapy, which was not allowed for ZUMA-1. Nevertheless, if you look at the response rate, 49% in the first one, 47% in the second one, which are comparable to the response rate from ZUMA-1. And duration of response is still early to assess, but 57% from the second experience is pretty promising, which is now in close proximity to the result from ZUMA-1. Other than the FDA-approved products, there are also several very exciting CAR-T products on their way to the regulatory pathway for FDA approval. This including Liso-cel JCAR-17 for B-cell lymphoma, KTX-19 for mantle cell lymphoma, and also the two BCMA CAR BB2121 and LCAR B38M for multiple myeloma. There are also more and more CAR-T products are being tested or entering early phase clinical trials. Specifically for T-cell lymphomas, now we begin to see CAR-T clinical trials aiming at targeting CD5, CD7, as well as TCR4. And there are also other novel targets for multiple myeloma, for example, CS1. For ALLs, the BAFF-R is being tested and is getting ready to entering into a clinical trial. So moving on to acute myeloid leukemia, there are only 40 clinical trials registered with clinicaltrial.gov. The main reason is being that acute myeloid leukemia, one of the major challenges for developing CAR-Ts or other engineered cell therapies for patients with AML is lack of targets that as pristine as CD19 for B cells or BCMA for multiple myeloma. An ideal target for AMLs would be a target that expresses on both leukemia blasts and leukemia stem cells, as well as this target will be shared by most, if it's not all, AML patients. And this target ideally should not express on any normal tissue or cells. However, despite continuous effort, such an ideal target has yet to be identified. And this is likely due to the clonal heterogeneity of leukemia stem cells between patients, and also the similarity between leukemia stem cells and normal hematopoietic stem cells, as well as lack of antigen with lineage-specific expression. So therefore, when we design a CAR target for leukemic, there's high risk for on-target and off-leukemic effect. Despite this, more and more targets are being tested in the preclinical setting. There's at least 40 of them nowadays, and several of them have entered early phase clinical trials. Louis White is the first one, CD33. There was a few case reports reporting on CD33 CAR for patients with AML, and most of our clinical trials actually focused on targeting CD123. NKG2D is another target that has been gaining more and more popularity. So there are four main CD123 clinical trials using CAR-T as the therapeutic. The first one is the one that opened at CTL Hope. It's an antiviral-based, either allogeneic or autologous CAR-T product, which has been active since 2016. Penn had a clinical trial initially using messenger RNA electroporation for transient expression. However, due to lack of activities, this trial was terminated in October 2017. Instead, in early February 2019, they opened a second CD123 CAR-T trial. This time, they use lentiviral CAR-Ts for persistent expression, and the study is early at its development. We're still waiting for the result to come out. Selectors using a universal CAR-T program and a lentiviral-based system to deliver CAR into off-the-shelf healthy donor T cells. And this trial was put on hold initially due to grade 5 toxicities. Subsequently, they reduced the intensity of the lymphodepletion regimen as well as the dose of the CAR-T, and the trial is currently ongoing. So this is the schema for CTL Hope's CD123 CAR-T clinical trial. For this clinical trial, there are two arms. Arm 1 is for patients with relapsed refractory AML. Arm 2 is for patients with BPDCN, which is a rare hematologic cancer, occur only in around 60 patients each year in the United States. And the study allows patients age 12 and above. All patients will need to have a donor or stem cell source identified. This is part of the mitigation strategy in case the patient, after receiving CAR-T treatment, develops an irreversible myeloablation. So far we are dose number 2 for patients with AML, As you can see here, all patients treated so far with AMLs, all of them have at least 1 or 2 prior allogeneic stem cell transplant. Majority of them received a CAR-T product made from their original donors. And we have seen pretty encouraging activities that we see patients with morphologic leukemia-free states. We have also seen a few patients with CR or CRI. In the BPDCN cohort, 2 patients were enrolled and treated. The first patient achieved complete remission at day 28. A second patient had MRD positive remission at day 28. So therefore, we think it's feasible to design CAR-T for AMLs. The current therapeutic response rate is encouraging. Of course, more work needs to be done to further optimize the CAR design, optimize the treatment planning in order to improve the response and also to benefit more patients. So moving on to the challenges for CAR T-cell therapies. The first one is for patients with highly aggressive disease. These patients will not be able to wait for CAR T-cell product to be made. They will require some sort of bridging therapy to stabilize their disease to avoid further complications. Therefore, a product with short manufacturing time is highly desirable. Currently, there are several products aiming at using off-the-shelf product in early phase clinical trials are ongoing right now. One of them was just recently presented at ASCO 2020 by Nilapo et al. This is the Alpha study, which uses ALLO-501. ALLO-501 is a CD19 CAR, which does not have the TCR expressed on the surface. It also does not have CD52 on the surface by using the TALEN technology. The lack of CD52 enables these cells to be immune to ALLO-647, which is an anti-CD52 antibody. This antibody is aimed to remove the host T cells to allow ALLO-501 to persist longer. So in these studies, the patient with DLBCL or follicular lymphoma in relapsed refractory setting received the ALLO-501 treatment. A total of 21 patients were enrolled. They were treated at various dose levels. 2 lymphodepletion regimens were tested with either a lower dose of ALLO-647 or a higher dose of ALLO-647. Remarkably, the median time for enrollment to start lymphodepletion is only 5 days. And if you look at the CAR-T expansion and persistence, not surprisingly, the patient with complete remission to the treatment has the highest peak of CAR-T expansion, the peak of expansion and also persistence. When patient received higher dose of ALLO-647, which is the anti-CD52 monoclonal antibodies, there's very nice suppression, the recovery of host T cells, allowing longer persistence of ALLO-501. 3 allogeneic off-the-shelf CARs are currently being tested in the clinic. The first one is the Alpha study. In these studies, the overall response rate is 63%. 37% patient has CRA, so 7 out of 19 patients responded to the treatment. The second one is summarized by Dr. Katie Vaswani's group from MD Anderson, which was recently published in New England Journal of Medicine, where they use CD19 NK. These NK products were derived from cord blood cells. In these studies, 11 patients were treated. 64% of patients, so 7 out of 11 patients achieved a complete remission. Precision Bio also had a PB CAR-19. This is CD19 CAR using the off-the-shelf product. 6 patients treated, 1 patient achieved complete remission, 4 out of 6 patients responded. It's still early. The number is small, but the response rate is quite encouraging. So far, all the allo CAR products appear to be safe with the encouraging activities. The immediate availability of the shelf feature of these products is definitely very attractive in the clinic, especially for patients with high-risk and highly aggressive disease. We're waiting for further report from the off-the-shelf CAR clinical trials for us to understand the duration of remission, and I think further study to optimize effective prevention of host versus graft rejection, allowing the allo CAR to appear to be necessary for a duration of response. The second challenge is using CAR T cells for patients in the clinic is what if patient with poor performance status and lack of reserve? In these patients, they might not be able to tolerate the second release syndrome and neurologic toxicity that are seen with the current FDA-approved CAR products. So for those patients, novel CAR design which preserve the efficacy but much reduce the cytokine production or neurologic toxicities is highly desirable. In the clinic, currently AUTO3 clinical trial is being conducted for patients with B-cell lymphoma. As of now, when they presented the data at ASCO 2020, no patient with grade 3 or above second release syndrome or neurologic toxicities, indicating possibly a better safety profile. And there are also efforts looking at optimizing the manufacturing platform to make the CAR secrete less cytokines but continue to be potent. On the clinical side, effective management of CRS neurotoxicities are also being conducted. The ZUMA-1, which including cohorts either given pre-antisteroids to prevent CRS or neurologic occurrence, or early use of steroids to reduce the severity of the toxicities. In their recent presentations, it appears that with these implementations, efficacy of axi-cel in patients with aggressive B-cell lymphoma maintained. And other efforts are testing using targeted reagents such as anakinra, dasatinib, seems to have anecdotal experience in controlling the toxicities. Last but not the least, when we are looking at patients who had CAR T-cell therapies, in this first case, the patient who had beautiful response to CAR Ts around day 40, unfortunately around 167, the disease came back. And the second patient, the patient did not have any response to CAR T treatment. So for these patients, efforts are devoting to improve the CAR T's fitness. So there are clinical trials looking at armor CAR, which try to potentiate CAR T activity by including cytokine production. And ibrutinib was tested in patients with CLL to potentiate T-cell function. PI3 kinase inhibitor were added to the manufacturing platform for the BB2121 with BCMA CAR. Other clinical trials looking at combination of IMiD plus a CD19 CAR as well as 4-1BB plus a CAR products are actively being pursued. Adding PD-1 and PD-L1 blockades in combination with CARs are also being tested in ZUMA-1, which is combining axi-cel together with atezolizumab, and platform study also looking at combining JCAR17 with durvalumab, and AUTO3 is looking at giving pembrolizumab prior to CAR T infusion in combination with a bispecific CAR against CD19 and CD22. And as we know that antigen or epitope escape has been quite common. It's seen around 50% of patients with ALL, around 25% of patients with non-Hodgkin lymphoma. And therefore, dual targeting, especially targeting CD19 and CD22, several clinical trials are looking at using dual targeting for this purpose. So what we have learned so far using CAR for patients with hematologic malignancies, we know that CAR T-cell therapy has changed the outcome of patients with hematologic malignancies. And we know that different CAR T designs and products are associated with different safety profiles. And definitely clinical expertise and established infrastructure are needed to deliver CAR T cells safely, effectively, and also to regulatory standard. An ongoing effort aiming to further improve the efficacy, reduce toxicities, reduce the cost, and potentially expand the indication to more patients. Last but not the least, cost effectiveness we're also being looking at, especially looking at those patients who receive CAR T therapies in long-term follow-up to see whether the CAR T therapy is cost effective. Thank you for your attention. Now I'm going to turn over the presentation to Dr. Preisman for his presentation.
Hello, everybody. Thank you, Elizabeth, and thank you, Mike, for the introduction earlier. That was a wonderful talk and an introduction to where CAR T cells have been used in the treatment of hematological malignancies. And with that, we've expanded as a field to try to treat solid tumors with the same effectiveness. And so that will be the topic of my talk today. These are my disclosures. And so Elizabeth did a wonderful introduction to CAR T-cell therapy. One of the major mechanisms that we take advantage of for the identification of virally infected cells or tumor cells is that T-cells can recognize a molecule on the surface of those cells called MHC. And in solid tumors and hematological tumors alike, that pathway can be shut off. So the normal recognition of those virally infected cells or cancer cells can be defective. And so that birthed chimeric antigen receptor technology, and Elizabeth went through this. I won't be redundant with that, but taking advantage of targeting cells in the absence of MHC presentation of peptides. And so our program at City of Hope is quite rich. This was a review we published many years ago now, but going through the production or manufacturing of CAR cells, which starts with leukapheresis of a patient, activation of T cells, and engineering. And in our program here at City of Hope, we use lentivirus for incorporating the chimeric antigen receptor molecule in T cells. They're expanded to billions of cells, and they're infused in most cases intravenously, as with hematological malignancies, as well as many solid tumors. And I think I'll present to you one example of how we at City of Hope have taken advantage of more regional delivery, and specifically in the context of treating brain tumors, brain metastasis in particular, where we infuse cells more regionally in the brain versus systemically. And as Elizabeth also went through, our program has treated a number of patients, the majority of which have been in the hematological space, and Elizabeth nicely went through her CD123 CAR trial for AML. We've had a lot of experience with treating glioblastoma, which is a solid tumor, as you can see here on the right side, and with that we've expanded to a number of solid tumor diseases. And I'll give you some vignettes of those today of how we've developed preclinically those CAR T cells and then translated them to an early phase 1 trial to treating patients with those T cells. And also Elizabeth went through a number of concepts that pertain to resistance mechanisms or challenges, and these are in particular facing solid tumors, tumor antigen heterogeneity. And most tumors are a mosaic if you think about that. And so if we target those with one targeted CAR T cell, there may be very easy ways for the tumor to resist that therapy. And so one can think of multi-targeted CAR T cell strategies, and that those come in various flavors with the idea that you can target multiple antigens that are expressed on the tumors, even in one mass, but certainly in metastatic disease where there are multiple lesions. We also think that CAR T cells may spark an endogenous immune response, and so while the instigator is CAR T cells, the anti-tumor activity one can see may spawn the host or the body's own immune system to fight the cancer after that. And also we can enhance that with various combination strategies, including other things that are also known to induce an immune response, like radiation therapy, chemotherapy, and I'll give you one example today preclinically of how we've developed a combination with oncolytic viruses to enhance the anti-tumor activity of CAR T cells. One of the other challenges that are facing solid tumors in particular versus hematological malignancies that also may have this strategy utilized by the tumor is to suppress an immune response that may happen in the tumor. We call that an immune suppressive tumor microenvironment, or TME. And we're also addressing those in various ways to try and overcome that microenvironment so that CAR T cells can work at their maximum capacity once they're inside the tumor for a potent anti-tumor response. And so another illustration of our CAR T cell program at City of Hope, where we started arguably on the right side treating a number of hematological malignancies, and on the left side treating brain tumors, and now we're sort of expanding into all other aggressive and at the moment late stage solid tumors. And I'll give you two vignettes on those, again, one in our brain metastasis program in treating patients primarily with HER2-positive breast cancer that have developed metastasis, and that's a very dismal prognosis in those patients. So in the literature, about 15 to 25% of breast cancer patients will be HER2 positive. And in up to 50% of those patients, because treatment strategies have been so effective in targeting HER2, that tumor cells find safe haven in the brain. And in 50%, up to 50% of those patients, they develop brain metastatic disease, which is very difficult to treat at the moment. And in prostate cancer, again, while early diagnostics and therapies have improved the lifespan of patients with prostate cancer, a good percentage of those patients ultimately develop what's called metastatic castration-resistant prostate cancer, also very difficult to treat with a poor survival, and we've developed a CAR-T cell targeting that disease too. And so, as I mentioned, route of administration, City of Hope was one of the first institutes to do this type of regional delivery, and this was on the heels of our glioblastoma experience, and I'm not showing that slide here, and I probably should have, but we treated a patient with a glioblastoma locally in the tumor bed, and that tumor never recurred, but he had multifocal disease elsewhere in the brain and down his spine. And we had preclinical data, and I'm going to show you some in the context of breast cancer brain metastasis, where we delivered cells not locally IC, intracranially, into the tumor, but rather intracerebroventricular, or ICV, into the ventricle, which is what houses the cerebrospinal fluid in the brain and the CNS. And when we did that, we actually showed that the responses were comparable to delivering the T cells directly into the brain tumor. And you can see that here in an orthotopic xenograft model where we're tracking tumors by firefly luciferase non-invasively. And you can see the firefly luciferase goes away in the brain, and the mice had prolonged survival. And one of the most important support data for regional intraventricular delivery versus systemic delivery was this piece of data with the same tumors injected in the brain of the mice, and when the CAR-T cells were delivered intracerebroventricularly, they cured every single animal. At a 10-fold higher dose delivered intravenous, we could only cure 50% of the animals, suggesting that regional delivery in this setting was much more robust in the anti-tumor activity than intravenous delivery. And we went on to show in this setting targeting HER2 that if there was multifocal disease specifically in the brain and in this example in the bone systemically, that CAR-T cells injected intraventricularly actually got to the brain, obviously, and to the bone outside of the brain at higher levels than when delivered intravenously. This was quite striking to us. We went back and did a therapy model and showed that intraventricular delivery of the CAR-T cells cured all the brain, as you can see in the middle bottom graph, and had more robust activity in the bone metastases and resulted in a much greater significant survival of those animals when delivered intraventricularly compared to IV. And so with that data and a lot of other data I'm not showing you, we recently opened a phase 1 clinical trial to treat patients with brain metastases or leptomeningeal disease with a HER2 targeting CAR-T cells, and in the CAR-T cell, and in this setting where intraventricular delivery, delivering these CAR-T cells to these patients. And this is a schematic of how the clinical trial is going, and hopefully we'll have some data to support this route of administration and this target for patients with brain metastases. And so in prostate cancer, we did a very similar thing. We went after a target called PSCA, or prostate stem cell antigen. And this is just an example, again, of our preclinical model of bone metastatic prostate cancer, where we injected these tumors. And actually, this is a patient-derived xenograft model of castration-resistant prostate cancer and injected into the bone of animals. And again, tracking those tumors by firefly luciferase. And in this case, we compared two different constructs. And Elizabeth shared some of the differences we've seen with CD19-directed CAR T cells for hematological malignancies. And in this case, we tested two different CAR constructs in the context of prostate cancer. And you can see there were striking differences. Both CARs eradicated disease early, but in one of the CARs in the third row here, you can see that in up to 80% of the animals, they recurred both locally and systemically, whereas the bottom panel shows in the second construct that it cured the animals durably in 90% of the mice treated. And in the bottom, you can see we tracked PSA, prostate-specific antigen, as a measure of tumor burden, and in the second construct, you can see there was no PSA detected in the serum. And this was largely in part because of the persistence of the CAR T cells, which was greater in the second construct. So with that data and also a lot of other data I'm not presenting, we've started a trial just recently treating patients with metastatic castration-resistant prostate cancer. And one of the things at City of Hope we take pride in is that we can expand this to other solid tumors, and this target, PSCA, is expressed in pancreatic cancer, also a very difficult to treat and oftentimes late diagnosed tumor type. And so hopefully we'll also follow up with that trial in the future. And as I mentioned before, the immune suppressive TME, or tumor microenvironment, has been a major difficulty in overcoming. And so to do this and to study that preclinically, we've actually built a better model system. So in all the models I showed you before, we inject human tumor cells into mice, and we inject human CAR T cells. In that setting, the mice have to have no immune system, essentially, so that they can accept human tumors and human T cells. So that does not allow us to evaluate the immune suppressive tumor microenvironment or other immune composition in the animals. And in this case, also doesn't allow us to test safety of CARs. And so prior to getting to trial, we actually built a fully immune competent mouse model so we could test the safety as well as the efficacy of our PSCA CARs. And I won't belabor the point, but on the bottom here, you can actually see that these animals express PSCA in normal tissues we know expresses PSCA in humans, including stomach, bladder, and the normal prostate. And so this may be sources of toxicities that we would like to evaluate in the mouse model as much as possible. And in this setting, it was quite interesting as well that unlike my prior slides, we saw no CAR T cell activity in this model. And so we really didn't have any other reason why other than an immune suppressive microenvironment. And so like in the hematological space where there's a preconditioning lymphodepleting step where we essentially make space for CAR T cells to migrate into lymphoid tissue as well as the tumor, we tried that here where we gave cyclophosphamide. I'm labeling it CPA, but it's an active chemotherapy agent we use for all of our CAR trials in hematological malignancies. And in this case, it had an anti-tumor effect by itself, but all the tumors recurred and no animals survived. When we combined it with our PSCA CAR T cells, actually 40 to 50% of the animals showed durable curative responses, which was striking to us. And one of the things we've looked at mechanistically is why. What is cyclophosphamide doing in the tumor specifically? And you can see here by histology of CD3 positive T cells that cyclophosphamide, and this is the third histology slide on this graph, on this page here, that cyclophosphamide alone allowed T cells to get in. So as I mentioned before, this immune suppressive tumor microenvironment prevents T cells from getting in. You can see that they're pretty much restricted to the outside of the tumor in the first two histology slides. And in the third slide with cyclophosphamide, they start getting into the tumor bed. And when we combined with CAR T cells, there was a striking infiltration of what we think were CAR T cells as well as host immune cells into the tumor. And we validated that by labeling the CAR T cells specifically with firefly luciferase and tracking them pre and post cyclophosphamide injection. And what you can appreciate here is that the CAR T cells had both early infiltration into the tumor and expanded to far greater levels, 2 log in some cases, compared to tumors treated with just CAR T-cell alone. And so we think there was a microenvironment shift with this lymphodepleting regimen, and we validated that by RNA sequencing, showing a number of genes, including immune signatures, that were dramatically increased or modified with the combination of CAR T-cells. And so another way to look at this is immunologically hot versus cold tumors, and one can think melanoma is an immunologically warm or hot tumor where there's an immune response happening in the tumor that's being blunted by the immune suppressive microenvironment and can be unleashed by things like checkpoint blockade, PD-1, PD-L1 blockade, and CTLA-4, whereas in prostate cancer and pancreatic cancer, we know they're typically immunologically cold. Checkpoint inhibitors don't typically work for that, and so we wonder whether CAR T-cells may. We've looked at this in pancreatic cancer models where T-cells are effectively excluded as the tumor grows, and this is just an example of that, where they're pretty much excluded from the tumor and are only in the peripheral sites. Whereas macrophages, on the other hand, that contribute to immune suppressive tumor microenvironments are infiltrating the tumors at high levels. And so to address that, we've looked at oncolytic viruses as a means to enhance immunity inside the tumor. This has been shown in a number of cases. In fact, there's an FDA-approved herpes simplex virus targeting metastatic melanoma called TVEC, and we know that it enhances immunity. We can also engineer those viruses to express genes of interest to increase CAR therapy. And our concept was very simple. It was on the heels of knowing that CD19 is an attractive target for CAR T-cells for hematological malignancies. And so we thought, well, in solid tumors where there isn't that type of unique and effective target like CD19, could we actually introduce CD19 into solid tumors and then target any solid tumor with a CD19 CAR? And that concept is illustrated here, where we engineer a tumor cell with this oncolytic virus to express CD19, and then we can go after any solid tumor with a CD19 CAR. This was a concept we tested actually preclinically. This is a fluorescence microscopy showing that when, in this case, triple-negative breast cancer cells are infected with the oncolytic virus, that they produce CD19 in purple, you can see expressed on the surface of these cells. When we combine that with the CAR T-cell, while the virus could kill tumor cells to a certain degree by itself, you can appreciate on the bottom in a time course in both triple-negative breast cancer on the left side and a glioblastoma line on the right side, there's a robust and synergistic anti-tumor activity with the combination of this oncolytic virus and CAR. So this shows that at least in vitro, we can deliver CD19 to a solid tumor and then target it with a CD19 CAR T-cell. We tested this in xenograft models where we showed, again, robust anti-tumor eradication with the combination with oncolytic viruses and CD19 CAR T-cells. And to again address safety of this approach as well as efficacy and looking at the immune microenvironment, we built a fully syngeneic immune-competent model to test this. It was very laborious for the graduate student who developed this in my lab. But we again tested this and showed durable curative responses in 50 to 60% of the animals treated with the combination versus it alone. And we actually looked at the microenvironment, so you can see the virus alone allows infiltration of T cells, which is hugely enhanced with the combination of CD19 CAR T cells. Again, as I showed before in another setting, we did RNA sequencing and showed a complete shift in the tumor in terms of its immune profile. And we actually proved preclinically that the virus was helping CAR T cells get to the tumor, again, tracking CAR T cells with firefly luciferase. You can see an enormous infiltration into the tumor of these CD19-directed CAR T cells. And what was maybe most promising about this is in the cured animals, we re-challenged them with tumor weeks later, and they were completely protected from tumor re-challenge, suggesting that there was an anti-tumor memory response that ensued following the combination of oncolytic virus and CAR T cells. So we were very excited about that. And so where do we go from here? We're obviously learning lessons from our phase 1 trials in prostate, brain metastasis. I didn't mention ovarian cancer or show data on that, or pancreatic pancreatic cancer, but we're certainly getting into those diseases. I showed some ways in overcoming the tumor antigen escape and immune suppression, and we're answering critical questions like how do we overcome this immune suppressive TME, and what's the optimal timing and duration of certain combination therapies, and how do we improve persistence and survival of T cells, and I gave you one example with oncolytic viruses, but this is certainly context and tumor type dependent, and we're addressing that. And so with that, I would love to acknowledge my lab in doing all of this work, the T Cell Therapeutics Research Laboratories program, which is directed by Steve Foreman, all of our collaborators within City of Hope as well as outside, and all of the funding that allows us to do this both preclinically as well as in our clinical trial. And with that, I'll give it back to Mike for poll questions. Thank you, everyone.
Thanks, Saul. And thank you, Elizabeth, Saul, and Al, for those informative presentations. We appreciate it. I'd now like to ask our audience to participate in 2 brief polling questions. Please do click directly on the screen to enter your answers. Okay, our first question, you see it up there. Which of the following best describes your experience with CAR-T cell therapy? There's 4 options: currently developing CAR-T cell therapy in the preclinical phase, currently developing CAR-T drugs in the clinical phase, interested in learning about CAR-T cell therapy, or developing other types of adoptive cell therapy. Once again, a question: which of the following best describes your experience with CAR-T cell therapy? Give the audience a few seconds to respond. Once again, the options: preclinical, clinical, interested in the area, or developing other types of cell therapy, adoptive cell therapy. As you see there, the results on the screen. Looks like a good portion of the audience is interested in learning about CAR-T cell therapy, 58%. Followed by a tie, 16.3%, currently developing a CAR-T drug in the clinic, or developing other types of adoptive cell therapy, both at 16% there. Thank you for participating in that question. We'd now like to ask our audience to participate in our second polling question. Again, please click directly on the screen to enter your answers. In which area is your main focus and interest related to CAR-T therapy? And then there's 3 options: hematology area, solid tumors, or both. Once again, in which area is your main focus and interest related to the development of CAR-T drugs? Hematology, solid tumors, or both. Give the audience a few seconds. And as you see the results on the screen, cancer definitely a huge area for this. So solid tumors, 40.4%. And it looks like companies have projects in both areas as well, in hematology and solid tumors, and 17% just hematology. Thank you. We'd definitely like to thank you for participating in our polling questions. Before we get started on the Q&A session, I would like to remind our audience how to submit questions. You can do so by typing them in the Q&A box, which could be found on the right-hand side of the presentation window. I'll now hand it over to Al.
Can everyone hear me?
Yes, we hear you. Thanks.
Okay,
Yes.
great. Okay, great. Thanks, Mike. Thank you so much. And thank you, Dr. Buddy, and thank you, Dr. Priceman, for providing excellent insight into the state of art regarding using CAR-T cell therapy for hematologic malignancies, as well as the potential for using this treatment modality, extending it to solid tumors. I also want to thank everyone in the audience for attending. I want to say that Precision for Medicine is really happy to host this webinar with applied clinical trials, because this treatment approach is especially important to the work that we do at Precision. I just want to say a few words about Precision for Medicine. We specialize in running clinical trials in which patients are identified through molecular diagnostic methods and trials that exploit a wide range of cutting-edge biomarker approaches to drive the development of immunotherapeutics and other molecularly targeted drugs. So cell therapy and other immunotherapies comprise a significant proportion of our ongoing programs. If you want to learn more about our capabilities, please download the cell therapy fact sheet that is posted here on the webinar's website. Okay, while our experts have been presenting, we've received some questions from the audience. So let's take a look at these questions. And the first question will go to Dr. Buddy. And the question is to talk a bit about the role of lymphodepletion in terms of benefits, risks, and how you make that decision.
This is Dr. Buddy. That's a great question. It really depends on the patient's condition and the disease the patient has. Now, the common use regimen nowadays contains fludarabine and cyclophosphamide. Now, this combo has been shown to not only to do lymphodeplete, meaning removing the regulatory T cells, myeloid-derived suppressor cells. They also have other functions, and they have been shown to increase the peak expansion, especially contribution from fludarabine. These are for patients with CD19 CAR-T cell therapies. So whether flu-cy or the dose that using flu-cy combinations, is that the right combo for other malignancies? No, it's not quite clear at this point. No, it's a commonly adopted combo, but I think for other solids, for example, solid tumors or even AMLs, no, flu-cy might not be the right the optimal combo. No, this really they have function not only to do lymphodepletion, but there's also function with regard to biological roles and whether they also contribute to to debulk the tumor and make the tumor microenvironment more amicable, more friendly to the infused T cells. And we also need to be very mindful that these are chemotherapy drugs. They have chemo-related side effects, and we know that from 2017 with the UCAR 123 clinical trials, the trial was put on hold initially because due to patient death, and one of the major contributing factors is the high intensity of flu-cy that was given prior to the CAR-T infusion. And recently, there's also a report from ASCO 2020 when the investigators using the ADP, A2M4 spare T cells with really high dose of fludarabine and cytoxan, and there were two related grade 5 patient deaths. Patient had aplastic anemia that developed after the T cell treatment, and there's also a patient with cerebrovascular accident. So subsequently, the intensity of the lymphodepletion combo would decrease in that trial. They have not seen further aplastic anemia. So I think that for lymphoma and acute lymphoblastic leukemia, it's pretty clear cytoxan and fludarabine are good combinations. But for other malignancies, more work needs to be done to really decide on the optimal combo.
Okay, great. Thanks for clarifying that. And now I have a question, a follow-up question for Dr. Priceman from your talk. The question is, with respect to intracranial administration, have you checked for chronic inflammation in the brain?
Yes, so that's a good question. Interestingly, we do, in certain cases, when we obtain biopsy tissue from patients pre and post therapy, we do look at inflammation. And we also interrogate the cerebrospinal fluid routinely because in the case of the intraventricular delivery, we have a catheter placed in the ventricle and can tap that tissue repeatedly, and we do look for inflammation. And I didn't present today, but we do see spikes in inflammation following the CAR-T cells, and we're trying to understand what that is a result of, and how does that play into the overall effectiveness of the CAR-T cells. So that is something we're looking at. In terms of unwanted inflammation, we don't see that in terms of cytokine release syndrome that has been shown in the context of hematological malignancies, and we don't see any to date, knock on wood, neurotoxicity.
Great, thanks for answering that. I have another question that could be answered by either one of you or both. The question is, have you used cryopreserved CAR-T cells for your studies, or were your studies utilizing fresh cells? And if you've used cryopreserved cells, what was the viability of the cells you saw on average, and what were there any notable differences between studies?
Elizabeth, so all of our studies use cryopreserved product, and we do significant release criteria on all patient products to understand viability upon thaw. Our criteria is that it's greater than 70% viable, but we're routinely over 90% in viability upon thaw and just prior to infusion.
Yeah, and for from the clinical side, no, even for the commercial products, Axicell, which is also called Yescarta, it uses and Kymriah, both cells are cryopreserved and shipped to the center once they are made. We have not seen a different efficacies when compared to patients receiving it under clinical trial, and there's also no differences among different centers. So I don't think that contribute to a difference with regard to the efficacy.
Okay, great. Thank you. Another question is, how do you ensure that the oncolytic virus specifically infects tumor and not healthy cells?
That's a great question. So typically oncolytic viruses and the majority of those that have been used preclinically or clinically have a tropism, a selectivity for tumor cells. That doesn't mean that they don't infect normal tissue, but safety has been shown widely with oncolytic viruses in the context of in clinical trials. In our studies preclinically, and what we plan to do clinically in the next year and a half to 2 years, is intratumorally inject. And in our preclinical studies, we showed that the virus was very selective in its infection of tumor cells. There wasn't any leakage of the virus outside into any of the normal tissues we evaluated alone or in combination with the CD19 CAR-T cells. So we anticipate great tumor selectivity of this virus. And as we'll show safety, hopefully, in our early phase 1 clinical trial, we do plan to move it out into different routes of administration, including regionally, if we're treating brain tumors or peritoneal carcinomatosis of various sorts. And we've done those studies preclinically. I just didn't show that data here. So we anticipate showing safety, and we can move it out to both regionally and possibly intravenous delivery.
Okay, great. Thank you. Here's another interesting question. What's your perspective on the potential of CAR-T cell therapy compared to other adoptive cell therapies for treating solid tumors, such as, for example, natural killer cell infusions?
Yeah, so, as Elizabeth presented, natural killer T cells have been shown to be clinically active now in hematological malignancies, as she presented. That came out of MD Anderson just recently. And there are a number of studies looking at, TCR T cells, TIL-based therapies, mostly out of the NCI, as well as NK cells. I think they all have great promise. perhaps equally, I think they need to be tested in specific diseases, maybe one more beneficial in certain disease settings than others. But they all show great promise. We're early on in our life cycle of testing all of these different cell-based therapies, and I think it's just an exciting time that we're in now.
Indeed. Well, it looks like we have a few other questions, but unfortunately, we've come to the end of a very interesting session. So I have to turn things back over to Mike, but I want to again thank everyone for dialing in. It's been a great presentation, and Precision has been thrilled to be part of it. So back over to you, Mike.
Sure. Thanks, Al. Appreciate it. Yeah, we'd like to thank the audience as well for attending and participating in today's event. I would also like to thank our speakers, Elizabeth, Saul, and Al. Precision for Medicine, and also Precision for Medicine for making today's educational webcast possible. You will receive an email alerting you to when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks to all for joining. We'll see you next time. Goodbye.